I'd like to thank everyone for coming, and I'd like to welcome you all to our panel on the current and future states of immunology. So this session, which is near and dear to my heart and hope it is to all of you, is going to explore the pivotal roles of the immune system in the development of various diseases, starting from some known areas and then really trying to stretch out into more novel areas. So first, I'd just like to quickly introduce our panelists. Okay, we have Dr. Alexandra-Chloé Villani, who is an assistant professor at Harvard Medical School and Massachusetts General Hospital. Dongfeng Chen, Dr. Dongfeng Chen, who is an associate scientist at MEI, an associate professor at HMS. Dr. Stephen Grinspoon, who is the chief of the Metabolism unit and a professor of medicine at HMS and MGH. And of course, my co-moderator today, Alec Stranahan from Bank of America. So I'm hoping to start kind of with a topic of immune system involvement that we probably are all pretty aware of at this point, and starting with you, Chloe. So your group has really been working to apply technologies of single-cell transcriptomics on human tissues to examine how changes in individual immune cells can lead to disease, okay? Now, why don't you tell us about what you've looked at and what disease your group has centered on so far?
Yeah, so kind of to help frame the conversation, we use cutting-edge technologies that combine the power of microscopy with the power of sequencing, where we can get the most granular definition of every single cell that make up a tissue to try to understand the cellular component and programs that drives disease. We particularly focus on the context of immunotherapy applied to cancer. And so we've been looking at the tumor bed, and I guess I can explain a bit more later on some of these adverse events we also study.
Yeah, and so just to place that also, so your group has been really trying to look at the precise cellular changes that are associated potentially with cancer immunotherapy.
Yeah, so it's been really fascinating to study human tissue through the lens of these technologies, which generate a really large amount of data to try to have a new look on how we could tackle and find new therapeutic targets in these environments. And cancer is a very fascinating disease because obviously every patient is unique, every cancer cells have their own specific makeup, but it's fascinating to see that this, regardless of which tumor patients have, their host immune response has so many common themes. I mean, the tumor bed is basically defined by unopposed immunosuppression, and that's consistent across all the tumors we're looking at, offering great therapeutic opportunities potential.
Fantastic.
Chloe, I was paneling a cancer innovation session yesterday, and the tumor microenvironment was one of the, I guess, top 3 areas of advancement being made in the treatment of cancer. How does the local tumor environment influence immune cell activation, infiltration, and does this maybe differ between different tissue types?
Yes, it's in a nutshell, to keep it simple, though I'm happy to go in greater details. The local microenvironment is capable of convincing our host immune system that they are wounded and that they should be protected. And so essentially shaping through different type of molecular mimicry the environment to sustain the growth. So you have a lot of immunosuppressive mechanisms that are in place. One of the ones that people have been talking a lot over the years is these immune checkpoints. These immune checkpoints are part of our normal mechanisms to prevent ourselves to attack self, to develop autoimmune disease. And these tumor cells have figured out a way to upregulate some of these molecules such that the immune system don't recognize them as outsider and thus don't attack the cancer cells. And so by figuring this out, there's been a whole slew of new therapies. These immune checkpoint inhibitor therapies, for example, that I study, are now approved for 96 different indications by the FDA. They're currently being used to treat more than 50% of all cancer patients in the United States. That is quite a revolution for solid malignancy. They're becoming the backbone of current trials.
So that's really, that's really fascinating, seeing the explosion of immune checkpoint blockade. But there's a lot of risks with this, right? Immune checkpoint blockade also comes with toxicities.
Yeah, so that's actually the main focus of my research. So these mechanisms Alex was asking me about, it's about relieving this immunosuppression. And I just mentioned in passing that these are key molecules that are there to protect ourselves from attacking ourselves, right, from developing autoimmune disease. So these are natural mechanisms. So it's deeply unnatural to target these molecules and to release the brakes. We release the brakes, and then the T cells can attack the tumor, and it's an amazing therapy before I get to the adverse events, because now suddenly, as opposed to having to tailor every treatment to every cancer patient where every tumor is unique, you have these common mechanisms that apply to all tumors regardless of your genetic makeup of your cancer. But it remains deeply unnatural to remove these brakes. So while you are attacking your tumor, suddenly we're seeing a lot of what we call immune adverse events, or IREs, that affect every single organs of the body. Actually, up to 90% of cancer patients develop these side effects. And up to 55% of them will develop severe grade 3, grade 4 side effects. So from deadly heart inflammation to skin rash. And these side effects are not going anywhere because these drugs are becoming the backbone of all combination therapy. In many ways, they actually mirror some autoimmune disease presentation. Clinically, they resemble autoimmune disease presentation. Sometimes we even try to treat them using some of the biologics that are available out there. So to us, there's an opportunity here to learn about human immune tolerance principles through the lens of these patients. Why do I say that? It's because when you try to study immune tolerance through the lens of autoimmune disease patients, using the technology that I use, and I could take patient specimens from patients that have Crohn's disease or multiple sclerosis or lupus, I don't know for how long the patient had the disease when I get the samples. Most of the time, they were on a diagnostic odyssey, so they were seen by many providers, receive all sorts of treatments, and when I studied their tissue, there's no way for me to figure out what's the driver from the bystanders of the disease. But in this particular case of adverse events, I know the trigger. I know when I gave the drug. I know the time point because I know when I gave the drug, and so suddenly we have a population of patients that we can follow over time, and we can see the emergence of these adverse events that clinically mirrors the early events of autoimmunity. And so if you remember only one thing from my remarks this morning is that as opposed to running away from these adverse events and shy away from them, there's an opportunity here to invest in studying them for a few reasons. First, for cancer patients, it is the path to improving patient care and quality of life, and if the first one to figure out how to mitigate them in parallel will be the one that will likely succeed in finding the next generation of combination therapy. Because these drugs are the backbone of many of the combination therapies out there. So these adverse events are not going anywhere. You need to mitigate them before you add up more layers of drugs. On the flip side, by studying them directly in human, you have an opportunity to define new drug targets tied to immune tolerance that could help a broad population of autoimmune disease patients. And so it can be a win-win for many different patients, which is why we decided to study them.
We saw a pretty interesting combination with Keytruda, or I guess PD-1 VEGF combination at, I think it was ESMO this past year. But this is kind of an exception, right? We've tried a lot of different combos on top of checkpoint inhibitors, and they haven't been able to render the survival benefits that I think we've been hoping, right, as an industry. Why do you think VEGF, specifically, if you've got thoughts on that, is a great partner with a PD-1? And maybe where are the other opportunities you see in terms of checkpoint combinations?
Well, it won't apply to all tumors because a lot of it, like VEGF, we're talking about, angiogenesis, and it depends very much on the tumor microenvironment. Not all tumor microenvironments are created equal. Some, even how immune cells are distributed, I'm taking a step back for a minute, even how immune cells are distributed across the tumor actually matters in terms of educating them to attack, whether they're outside of the tumor, where you have something more cold inside of the tumor, and then high hypoxics. If you have a lot of hypoxic, if you have a lot of vascularization, will actually influence if you're gonna respond to this type of treatment. So some of the efforts that are being done on an academic setting include some of these large tumor cell atlas initiative where we're using the type of technology that Carrie introduced me through the single-cell multi-omics strategy to try to characterize more granularly which tumors and which patients would benefit most from these drugs. And very often right now, the way it's done is we don't take this into account when we decide which patients should go for which drug regimen. That probably influence some of these outcomes because there's only a subset of patients that would really benefit from targeting vascularization and hypoxic signature.
And that's so interesting, that topic, because you introduced, at some point you mentioned, you introduced the concept of precision medicine, which we're all quite aware of and which we think of a lot. And precision medicine, when it comes to tumors, we think about it as really identifying the genetic defect that's leading to that tumor. And what's interesting so far about the way that we've thought about immunotherapy is that it isn't very precision-based, right? We're using the same immunotherapeutics for people regardless of what's going on in their tumor. Do you think that potentially that might be changing, or that we're on the cusp of precision-based immunotherapies?
It's interesting you say that because, half of my work is on autoimmunity, and if you look at it through the lens of autoimmune disease, they tend to say, oh, in cancer world, you are doing precision medicine. So it's a question of perspective. I think it's going to be a mix of both. Like cellular therapy is a form of precision medicine, and when it works, because you are re-engineering a cancer patient cells to attack. It is very expensive to do precision medicine at scale and honestly not really cost efficient from an economic standpoint. So you kind of need a combination of off-the-shelf solutions, which really Checkpoint provides you, with some tailored therapies for some of the patients that don't respond. So yes, Checkpoint, we were lucky in that these are broad principles that are conserved across all tumors. I think there's been a big race after the first checkpoints were discovered, and with the hope that we'd discover more of them. There's LAG-3 now that is out there, and some of these other targets have not been as promising. So yeah, as we learn more about these tumors, perhaps it won't be so much precision medicine as of tailoring the treatment for every patient, unless you do cellular therapy, but more in terms of defining which other pathways are on, like I was mentioning VEGF, in which patients, and trying to kind of tailor it this way.
Okay, well, your work has shown so much about the dual sides of the immune system, in both immunosuppression, needing to overcome that within the tumor and microenvironment, but then also kind of revving it up and leading to potentially autoimmune diseases. And so now I want to turn to Dr. Grinspoon's work. Steve, your work has really focused on how kind of chronic, but even potentially local alterations. So we're not even talking systemic, we're even talking about local areas or alterations in immune function can impact the entire body, okay? So especially when it comes to metabolism, especially when it comes to, both cardiovascular disease and endocrine disease. So I was wondering, what are some of the metabolic diseases that are influenced by the immune system, and how does this happen?
So thank you. So our lab, we study ectopic adipose tissue, and we study models of obesity in which there's excess fat in the visceral area. And when people think about obesity, they tend to think people are overweight and all fat is the same, and that couldn't be further from the truth. And in fact, the visceral fat is really the fat that leads to increased inflammation, and the subcutaneous fat, which is around the hips and the buttocks, is actually very protective, and it's a very important, metabolic sink that's protective. So what we've studied is selective strategies to reduce visceral adipose tissue. And in the visceral adipose tissue, as you were mentioning, there are crown-like structures, and that tissue increases innate immune activation through IL-6 and other pathways. And in fact, modulating it specifically in being either neutral to subcutaneous fat or actually increasing subcutaneous fat, making it more functional, actually improves inflammation. And so the current strategies lower both visceral and subcutaneous fat, and they lower inflammation, but I think ultimately drugs such as we've developed might be more targeted to reduce inflammation. I'd also like to add that inflammation is an ancient system. It was developed to kill pathogens and fight infectious diseases. So it's an excellent, important thing, okay? And interestingly, in the days of tuberculosis and all those precursor times, the people who were the thinnest died, and people who had higher weight survived. And it's hypothesized that that's because they were able to mount an inflammatory response to the pathogen. And even things like insulin resistance, could actually increase glucose to the brain because you're insulin resistant. So this whole set of conditions and diseases that we call metabolic syndrome and obesity developed as kind of a protective way to fight pathogens. And the problem now is in this era of food and concentrated sugars and whatnot. We've gone from the healthy part of the U-shaped curve to the other end, in which this system is turned on its head and is out of control right now. So I think there are various different ways to modulate these depots and through diet and other strategies. And we've also looked at some strategies to reduce innate immune activation and cardiovascular disease, which I'm happy to talk about as well.
Steve, I'd be interested to hear your thoughts, given the outrageous demand for GLP-1s and weight loss. And I mean, obesity has been a pandemic. 60% or more, I believe, of Americans are overweight or obese. So the demand for these drugs has been unprecedented. I think even the GLP-1 manufacturers have been surprised themselves. But do we know what happens after the weight's lost in terms of the impact, on muscle or on the immune system and sort of the interaction between that loss of fat and how the body reacts?
So the muscle is a really important point to discuss, and it's really a little bit of an Achilles heel to these GLP-1 agonists and others, because there is significant muscle loss that's associated with the weight loss in general. And muscle, as you know, is important to maintain glucose and functionality. And so I think ultimately there will be combinatorial strategies which increase muscle at the same time as fat is lost, and you can do that through modulation of the activin system or other pathways, and that's really important. I think in terms of inflammation, I think as people lose weight, their inflammation will be modulated and stuff. I don't think it's going to be completely turned off, and I don't think these drugs are necessarily anti-inflammatory specifically. They're just secondarily anti-inflammatory because of the weight loss, though that is an open question. And so, for example, the fact that these drugs reduce heart disease is really, really interesting. And is it simply because of the weight loss, or is it because of some immune modulation? I think those are important open questions. And if it's the latter, maybe that could be targeted independently of the weight loss. And so that's a really important question.
Yeah, I mean, that really kind of gets at the central question with immunometabolic diseases, the chicken or the egg question, right? So, can we treat these by targeting inflammation, or do we have to target them by weight loss, by metabolic processes? What do you think is, we hear a lot about how we live in a pro-inflammatory or a high inflammatory state. Which is driving which, and does it matter?
Yeah, it does matter, and I think treating inflammation to reduce, for example, cardiovascular disease, that's really hard, okay? And this has been a tough nut for the biotech industry to crack. And I'll tell you a few tidbits regarding that. So first of all, it is true that when you reduce inflammation, you reduce cardiovascular disease. In the CANTOS trial with an anti-interleukin-1 beta showed that, because that study in people that had inflammation and heart disease without lowering cholesterol lowered inflammation. The problem is, because of what I said before, because it's such an ancient system that's so important, if you turn it down too much, you will get infections. And that is exactly what happened in CANTOS. They had increased fatal infections. So finding the sweet spot is not that easy. Colchicine is now being developed as a tubulin modulator. It affects the inflammasome and neutrophil function. And has had some luck, and it may be something that is on the horizon. Our group recently looked at statins. People think about statins as they reduce cholesterol, and they're wonderful for that. But actually, they have serious anti-inflammatory effects. They reduce the inflammasome, they reduce IL-6, they reduce innate immune activation. And we just did a really big global study in which we gave statins to people who met no indications for statins. These are HIV patients with chronic innate immune activation. This was the Reprieve trial recently published. And the effect on cardiovascular disease was twice the level of what the reduction in LDL would have predicted. And the other half is probably some modulation of immune function. And we've shown, we're looking at the genetic mechanisms of that right now. So statins, ironically, because they're not as strong as canakinumab or something, they don't lead to increased infections and stuff. So they might hit kind of a sweeter spot. But I think, so I think, for the patients, it's always better to start earlier, like what's driving this? If it's obesity, that's important to treat. But for others, who have, who you want to do cardiovascular prevention for, who you're already trying to treat the obesity, targeting inflammation will be a major focus in the future.
We've talked a lot about shutting down the immune response, but there's also safety aspects on the other side, right, of potentiating the immune response. I think we've seen with IL-2s, right, they've carried their own safety issues, and there's companies now developing extended half-life, less potent, but in the body for a longer period of time. So maybe just speak to the other side of that equation, right? And how do we strike this balance? Where's the sweet spot?
Yeah, as I mentioned before, I think that you need some degree of innate immune activation that's ongoing for pathogen hunting and prevention of fatal infection. And so I think turning down that system too much is really a problem. I think strategies which act, for example, more locally on coronary plaque to reduce lipid oxidation and other more localized processes are probably the ones that are going to be the most successful. Because lipids, when they get into the coronary plaque, are themselves inflammatory. And so, that's why statins are sort of doubly effective. They reduce lipids, and they reduce the entry of lipids, and that further reduces the localized inflammatory microenvironment without a mega effect systemically to turn down inflammation. And there may even be targeting of strategies to the coronaries, I could imagine in the future, that specifically modulate these pathways. So anything that's what causes heart disease, if you will, it's localized inflammation. Of course, inflammation can affect the kidneys and it can affect multiple organs, but I think in terms of preventing major adverse cardiovascular events, local strategies would be most useful.
Yeah, and that really kind of gets to kind of local versus long distance, how the immune system can be communicating with the body as a whole. And it's an interesting question, especially for Dr. Chen's work, I think, because Dr. Chen, you're looking at neurodegeneration, you've looked at the eye, you've looked at the central nervous system. Now, these are supposed to be immune-privileged sites, right? So how have we gotten that wrong?
Yeah, thank you for the question. And I would say that when we first realized the roles that how immune system play in neurodegenerative process, it was first a surprise to us. So my lab uses eye as or especially retina as a model system to study CNS degeneration. So we all know that retina or the brain neurons are actually sheltered behind this blood-brain barrier and blood-retinal barrier. So they're supposed to be protected from the T cells or peripheral immune attack. But then when we started to use the glaucoma, which is the most frequent neurodegenerative degenerative diseases in our body, actually far more frequent than Alzheimer's disease. So we were surprised to find that there are actually T cells coming into here. And we thought they're supposed to be shielded from the T cells. And then we realized not only the T cells comes in, it's actually participating critically in the disease process. So if we use the, let's say, mice deficient for T cells, and then you have much attenuated neuronal loss. And also, if we isolate T cells from glaucoma mice, and some other labs actually even isolate glaucoma T cells from glaucoma patients and transfer it to a normal mice, and this mice develop glaucoma. And I'm really saying that our blood-brain barrier, blood-retinal barrier is never that tight. It actually could be leaky at some, I think especially for activated T cells, they actually have the ability to penetrate, to come into the retina and the brain. And so later, there's more papers reporting that T cells also comes in the Alzheimer's disease patient and participating in neurodegeneration in the brain. Therefore, now we realize glaucoma is no longer just a localized disease. It's a systematic immune response actually contributing to the disease process. And so talking about this, and when we published this work, and we got patients who write letters to us and also to our glaucoma specialists and say, sharing their experience about how they have autoimmune conditions or infections exacerbate the disease of glaucoma or vision loss. And therefore, the glaucoma specialists in our department came to collaborate with us and say, well, that's partly looking at whether the same process is really happening in humans, and we're not only looking at the mouse model. So we actually identified using this like a trend of thoughts and really identified first liquid, I would say peripheral blood biomarkers that could show quantitative correlation with nerve degeneration in glaucoma. So, that's why I said it was a surprise, but it was really fundamentally underscore the like immune mechanisms in driving the neurodegeneration.
Yeah, I want to follow up on that point around biomarkers with cognitive decline. I think we're seeing more and more this is becoming very important in diseases such as Alzheimer's, where you don't want to necessarily give continuous treatment. There's a new drug, donanemab, that's been approved by Lilly, where you essentially reduce the plaques to zero and then you go off treatment. But I still think we don't have a full picture of the biomarkers, even in that very well-studied disease. So I'd be interested to hear your thoughts on, do we need to find specific biomarkers for each degenerative condition, or is there maybe a broader biomarker of cognitive decline that we should be looking at?
Very good point. This is actually part of the questions we're asking ourselves. And while we identified this blood biomarkers, first one in glaucoma, we were wondering whether that's the same biomarker shared by other neurodegenerative diseases. So we're actually get a grant and now checking in the Alzheimer's disease patient blood and to see whether there's the same T cell biomarkers or actually their T cells probably targeting a different antigens or pathogenic antigens in the eye and the brain. But in either conditions, I think there is a possibility that brain and eyes probably share different kind of antigens. They haven't been exposed to the peripheral immune system, and that's why when the blood-brain barrier is leaky, then T cells comes in and starting to attack those specific antigens that the first time they've seen in the brain and retina. So we do not know, but at least we think that really provide a gateway for us to both to assess what kind of patients are likely to be sensitive to this immune therapy or have underlying cause of the immune therapy, as well as using it as a measure for earlier measure to predict the therapeutic potential efficacy.
You know, Dr. Chen, often when I think about inflammation in the central nervous system, I'm thinking about microglia playing a role. These are the resident immune cells of or type of immune cell within the central nervous system. Are they interacting with these T cells that you're talking about? Are the T cells changing their state? And if so, who do we target? The microglia? Do we target the T cells? Is this reversible?
You really asked the key point here. So And actually, when we realized the T cells coming to retina, and one of the key questions we're asking is how? How does T cell knows to come into the retina? And then we come to realize that the microglia is the residence macrophage in the retina, actually the key drivers. They are the one who first sense the stress or damage inside the retina. And then they open up the door for T cells and attract them to come in. So when we use, let's say, antibodies or to calm down or to flatten the inflammatory response of microglia, and what actually comes down like attenuated T cell to comes in. So it's all started from the microglia, and that is the key drivers that drives the entire immune responses in the retina and the brain. So then we're talking about what activated microglia, right? And I think there's so much more knowledge about what activates microglia, or almost we can say everything activates microglia. Pathogen-associated molecular pattern, we call PAMs, DAMPs, damage-associated molecular patterns. And now people specify and say neural damage-associated, I guess, NAMPs. But really the key is we know very little about what turns off, what the cause of the activated microglia. So the reason we come to this, and also when Steve was talking about, to manage the inflammation is actually the key. I mean, like we don't want to, the inflammation is double-sword edge. You don't want to shut it down. But actually natural already have this built-in mechanism to calling the inflammation off when the damage is neutralized. And that is actually leading to a smooth process of, because this double-edged sword is not only to defend while it's causing damage, but also there's another process that follows with this defending process is to promoting tissue wound healing and also repair and regeneration. So I think that smooth transition has to be there. If we just simply inhibit or suppress inflammation, then the wound is still there, the debris is still there, and the damaged cells are still there. If nobody cleared them, and when you remove the suppression of inhibition of inflammations, those wounds will still be activated again and to trigger a new round of inflammation. That's how we understand of this process.
Yeah, I mean, this seems to be a trend that we're hearing from Steve and also from you, Feng, is it's an ancient system that we're still learning about, right? And we're finding that the immune system can go places that we didn't necessarily thought were possible or even biologically needed, right? So how do you see this progress evolving? Are we accelerating in terms of our understanding of the immune system? And are there still sort of areas of unmet research?
Yeah, no, that's, so when we thought about this, I mean, really one of the effort is we want to find that natural developed resolvers, or we call it that actually calling off the inflammation, because that will lead us to much more balanced suppression or guidance of turning off the immune system. And one of the really exciting discovery in the lab is we actually found one of such molecules it actually turns on after like a few weeks when neuroinflammation occurs in the retina. And we found out what's really interesting is we found out that this molecule, when we take this molecule off, let's say knockout, right, in the mice, you completely delete the genes, this mice will spontaneously develop glaucoma and also early manifestation of Alzheimer's disease, which means brain topsy. It really underlines the cause and supporting that inflammation or microglia activation is the drive of neurodegenerative disease. It's not just a bystander. So it's the underlying cause of it. And however, we also noticed that if we inject these molecules back to the glaucoma, it has such a potent neuroprotective effect that I've never seen when we tried neurotrophic factors or immune suppressors. So again, in saying that it's important to drive a balanced return of the immune process, and that probably will present much more effective therapy and also control of neurodegeneration.
Now, I mean, this balance obviously is so important and so hard to strike. But what I find really interesting is that each of our panelists, each of you, have spoken about the need for balance, okay? And each of you have spoken about how when that balance is lost, we can have immune-related adverse events, which could be cardiomyositis, which could be which could be, but in very specific locations and different locations per different people, right? Now, you, Dr. Chen, just spoke about, the eye and the central nervous system, neurodegeneration as a site of this loss of balance. And Dr. Grinspoon, speaking about the cardiovascular health, what strikes me is that you guys have all indicated different organ systems ultimately that are targeted by loss of this balance, and yet each of them is potentially related to the same imbalance, right? The pro-inflammatory state. So I wonder for the panelists, what do you think leads to this specificity, okay? Or if we have no idea, what do we need to do next? How do we need to look forward as a field to understand what the central issue is, but really what's affecting our individual patients?
One thing I didn't add before, which I think is a whole other level of complexity, is the microbiome. And so, we all have this whole ecosystem inside us that's, so many species and ones we don't even understand or know about yet. But, and you hinted at this, but, the gut contains a lot of bacteria, and the types of bacteria and the fermentation and the different processes are really, really important. And what you eat, especially sugar or fats, can actually have, or how much you weigh, can have a serious effect on that. But even more, we're starting to understand that it can get leaky at times. And there's actually a whole nother way to thinking about inflammation and modulating it is to, affect the microbiome either through, in dietary strategies, or even more interestingly, through strategies like GLP-2 agonists, which affect gut leakiness, which actually decrease the bacterial extravasation from the gut. So, I mean, in our world, we're trying to tackle, weight, but also these other systems which all contribute to inflammation. And I think it's really, really complicated, and I think we're going to have to think on multiple levels in this regard.
Well, if I would add, that actually reminded me that there's one really interesting finding when we studied the glaucoma, and we found that these mice that we call germ-free mice, that means mice never exposed to germs, and which means their microglia and macrophages, probably even T cells, not being primed by the bacteria. And so it seems like the microglia are not ready to respond to lots of the injuries, or they stay quiescent. In these mice, they don't develop glaucoma. So that's, I guess that's really another evidence, or actually the first evidence made us realize why microglia is the driver and the neuroinflammation is the driver of this whole neurodegenerative process. And but as we studying further into this, and we realized this is a systematic regulation, a metabolism regulations, like gut bacteria, is releasing certain metabolites that actually can, I would say, distantly regulated the inflammatory response and the microglial response, even in adults, in our when we're at this stage. It doesn't have to be like through the development.
So you ask us how we can improve, care, and I will definitely say that in the context of the pathogens that I study, microbiome definitely is a modifier, not necessarily a driver in the context, but a modifier, which we should not ignore. I'm a big so there's so much more to understand about this balance and what tilts it and how to get it back right. And a lot of our so I'm a big proponent of doing more human studies in tissues, because while we do need these preclinical models, they're essential to develop these drugs and test them. Some of these principles don't translate so well back and forth between mouse and human. You know, we've cured our preclinical models thousands of times from tumors. Interestingly enough, none of them develop adverse events aside from vitiligo. So clearly there's some distinctions with human. And the question in my field is always, who picks up the bills for these in-depth, expensive analysis of human tissue that are so rich in information in identifying new therapeutic targets that we could then try to see what's conserved in animal models? And it's like the hot potato game, you know? It's like, it's not NIH that cannot necessarily fund this, it's not private foundations, industry, of course you have your own agenda, and so there's a need to rally more in terms of identifying the right cohorts of patients and tissue. It's not a catch-all, and like doing more in-depth analysis so we can create these more complex understanding of what's going on. I'll be a bit more specific. I talked about adverse events. Clinically, they may all look the same when they come in the clinic. Phenotypically, in terms of, you know, your patients has diarrhea, has colitis, or, you know, some arrhythmia, maybe myocarditis. But when you start looking molecularly, a few patients that look the same are there for different reasons. In the context of adverse events, it can be the reactivation of resident memory T cells that were there all along, and we have proof of that. But in the same tissue, it can actually be autoreactive T cells that are responding to shared antigens with a tumor. It can also be complement activation. It can also be tied to autoantibodies, and they all look the same when they go through the doors in the clinic. So we need to figure out which is which, because you should not all be treating them the same way to rebalance them. And we're only starting to unravel this complexity. And this is, of course, in the context of immune adverse events, which is somehow a more controlled environment, because we know this patient population not having autoimmune disease for now, in terms of we haven't been treating so many autoimmune disease patients with these drugs, and we know the timing and the drugs being used, and even that we're seeing this complexity. And you can only imagine how that complexity translates to neurodegeneration, metabolic disease, and autoimmune disease. And we kind of need to find a way to better team up to kind of tackle this from the human side.
That's true. That's true. Now we have a few more minutes left in the panel where I'd like to open it up to the audience if there are any questions. I know we have someone around with a mic in case anyone has questions.
There's one here.
How could we measure the induced T cell response in people?
So when we are looking at the blood biomarkers, we take the peripheral blood cells, and normally we have to know which antigen is responded to. So we use specific antigen to stimulate the cultured T cells, and then looking at the T cell response. And you can compare, let's say, diseased patients and normal populations, how these T cells respond to different specific antigens. Does that make sense?
I mean, to the broader question of how do you identify inflammation that's predictive of disease, which is a question I deal with all the time, particularly with cardiovascular disease, we're at our infancy. I mean, honestly, we use things like CRP, which are just like a SED rate. It's just like there's something happening. We don't even, but there was, a huge paper that was published a couple weeks ago showing that in people, the level of that marker 30 years prior predicts disease, 30 years into the future when you control for cholesterol and other things. So I think CRP itself is not a disease causer, but it is a marker. And so I think it's incumbent on us to understand better these markers, because you could target, and that's another point we haven't talked about. We're talking about throwing anti-inflammatory strategies on different people, but they really only should be targeted to the people who need them, who are inflamed, who are most at risk for this disease. And so I think a huge effort needs to be made to understand markers that predict inflammatory comorbidities, if you will.
I think we've got one more quick question here.
There's an increasing body of evidence that omega-3s that they have immune modulatory propensities. So they're not only downregulating apparently immune system, but they're also stabilizing it. Can you say something about it? What's your opinion?
I think that's absolutely true. I think whereas saturated fats and other things are the opposite, and high sugars, high concentrated fructose and other things increase that. Omega-3 fatty acids absolutely decrease that. And foods that contain those kind of things are extremely healthy. And diets that are balanced in anti-inflammatory food. There was just a great article in the New York Times this morning, if you will, in the health section, which I just read, which actually goes through a lot of different types of foods, and there's a whole section on omega-3 fatty acids, to your point. But there's other foods, fibers and other things like that are also anti-inflammatory. There's certain fruits and vegetables have antioxidants in them and stuff. So I think, the Mediterranean diet is probably successful because it's an anti-inflammatory diet. So I do think what we eat would really, really help in terms of the long run. So it's a great question.
One more question.
Hi, so you talked about how you've induced, adverse events. Have you studied or applied, what you've learned through that process to how the body triggers an autoimmune disease sort of organically?
Yeah, that's an excellent question. Thank you for your question. That's a bit what we're trying to get at through the lens of studying these adverse events in patients that don't have autoimmune disease family history. And you're asking if we've advanced it. I think we're a little early in the days of having fully figured it out. It is clear that it's not as simple as genetic modifier or everybody walking around with specific type of autoantibodies or reacting the same antigens. That it is a bit more complex. I think one of the reasons we haven't made great advance is that it's actually really hard to convince people to work on this and to fund this, to be honest, because it's the flips. Everybody wants to think about the next drug targets, the next combination, improving efficacy, improving non-responders. So sitting on this panel and hoping to convince all of you that it's actually a valuable field to embrace and study, because you could really crack open some of these principles for autoimmune disease patients, in addition to kind of helping with a roadmap of therapeutic targets for cancer patients. And so I don't have a straight answer for you here, because the field is just starting. I've been in it for 7 years, and it's been a little bit of a lonely venture. So I hope a lot of you will support this venture and join us on it, because there could be some solutions right there.
Well, I think that's a great point to end on. So I want you to join us in thanking our panelists for the great conversation.